Howdy. I know post-lunch session could be one of the toughest of the day, but yeah, the next talk will be exciting, I'm sure. So we're starting our afternoon session with Dr. James Schnable, the Nebraska Corn Chekhov Presidential Chair at the University of Nebraska, Lincoln. Dr. Schnable leads a distinguished research program at the intersection of quantitative genetics and machine learning, focusing on improving the agronomic performance of maize and sorghum. In addition to his academic work, he is a successful entrepreneur in the fields of bioinformatics and precision agronomy. A graduate of Cornell and UC Berkeley, his contributions have been recognized by the Maize Genetics Cooperation and the American Society of Plant Biologists. Please join me in welcoming Dr. James Schnable.
Howdy. That's really fun. All right, so I want to thank the organizers. This conference has been really well organized. I went to one of these a couple of years ago, and another speaker pointed out the students, I think they take this much more seriously than a lot of faculty do when we have to organize, and it shows. So thank you for that. And thank you also for the vote of confidence that comes with putting me right after lunch. That means that it's my job not to have you folks doze off in the next 45 minutes, so we'll do the best that we can there. I think this is actually presented. Oh, okay. Yeah, perfect. All right. So before I get started, there's a disclaimer down here. As you heard, in addition to my professor hat, I do some other work as well. If you want to read a lot more about it, there's a whole page about that. If you want the quick version, here's a whole bunch of logos. Of these, the only conflict that I think we're actually going to touch on at all today is dryland genetics. It'll be very obvious. It's the first couple of slides. So in the morning sessions, I think we heard a lot about what new technologies are allowing computers and drones and image analysis to do better than a human being. We also heard a little bit about what humans can still do a lot better than a computer. And that's one of the great things about the work I get to do, is to see see that line and see where it's moving. I'm going to spend a lot of time talking about new approaches to image analysis. So I want to start off with something that humans are absolutely better than computers at, even today, and something that, it's one of the real factors that has enabled our progress in plant breeding and genetics. And that is scoring early stage yield potential. So this is my conflict of interest. This is Dryland Genetics. It's a company that breeds proso millet, crop grown about 500,000 acres in Nebraska and Colorado. You guys have one of the most successful proso millet breeders on the staff here at Texas A&M. But Santosh is a graduate of University of Nebraska. He's employee number 1 at Dryland Genetics. And what he's doing in this photo is he's walking through a large field of about 10,000 varieties of proso millet that have never been seen in the field before. Just, a recombination from different parents. And just looking at these varieties, he's making the determination that 90 to 95% of them will not move past this stage of trial. They will not go beyond a single several foot long plot of seed into future replicated yield trials. And I don't really know how he makes those determinations, but they work. We've had the chance to, take stuff that he picked and take stuff that he didn't pick. And the ones that he selects do much, much better. This is what we heard about in the morning, the breeder's eye. And it's hard to find direct evaluation of this. The closest studies I have found come out of Africa, and they look at something called farmer appreciation. So you grow a single plot of some variety, many varieties. You bring farmers in and you just ask, which ones look good? Which ones would you like to grow? And then the next year, when you have a lot more seed, you can go out, you can do replicated yield trials, you can get meaningful yield data. And the farmer appreciation scores are better predictors than the actual measured yield of which varieties will do well in those replicated trials. So what's going on here? Yield is actually a pretty noisy trait, and particularly if you're looking at a relatively small plot. So if you are going to have noisy yield data that can be thrown off by variation in soil conditions, variation, a raccoon came into the field and pulled a few ears off of one plot. And you can also look at lots of other traits that are correlated with yield, but are more highly heritable. You can do a pretty good job of predicting which varieties will do well in future environments. And the really best breeders have learned how to do that. Usually they can't tell you what they're looking at. They're looking at the field and they're saying, this one looks good, this one doesn't look good. But for most of the history of plant breeding, this initial visual evaluation has underlined a huge proportion of our genetic gains. And even today, outside of 4 or 5 major crops like maize, like like soybeans, like wheat, where we have really powerful genomic prediction methods, this still is the first step in the pipeline for many breeding approaches. So, yeah. We have a problem, though, which is there aren't enough people like Santosh Rajput in the world, and we're not making enough of them. And realistically, that's not going to change. Think about what goes into making a skilled plant breeder. 4 degrees of undergrad, probably 5 to 7 years of graduate training. Then you get hired, you go out to a breeding station somewhere, you think you have it made, you get sort of 3 or 4 or 5 years in, and sometimes everything's great, and sometimes you find you don't have that job anymore. Because part of what's happening in those 3 to 5 years is an evaluation. Do you really have this ability, this breeder's eye? So the breeders we have are outstanding, but to what extent can we figure out what they're looking at when they're walking through the field and making those evaluations? So this is work from Ravi Morale, now a professor at South Dakota State. This was a collaboration with Addie Thompson's group at Michigan State University. So we had a really large maize panel evaluated in 2 environments with some yield data. And one of my former graduate students, Hong Yu Jin, took this the data on maize performance. He also took data on essentially everything we could think of to measure up through flowering time. So anything you can walk through a field with a ruler and a notebook and make measurements. And he built 2 models. One model was a straightforward genomic prediction model. The other is what we call a phenotypic prediction model, which gets a little confusing because I know Seth has phenomic prediction. The difference here is we're looking at specific discrete traits, not patterns in the overall data coming off of the sensors. And what he found is in this particular population, the phenotypic prediction roughly matched the performance of genomic prediction when we looked at cross-validation within an environment. And it somewhat outperformed, again, in this population, which is pretty diverse. It's probably not the best proxy for advanced breeding lines where you have more kinship and more population structure. But it outperformed genomic prediction when we wanted to predict performance across environment from phenotypes measured in a single environment, just measured in Nebraska. The other nice thing about this is a simple random forest model. This is not a really complex deep neural network, so it's straightforward. We can go in and pull out what are the factors the model's paying the most attention to. When we do, we see things that make sense. So flowering time matters a lot for yield, matters a lot for adaptation, and the optimal flowering time is different in Nebraska and in Michigan. There are other things that make a little bit less sense, and I think are one of the reasons it is important to always take the step of digging into why your model's getting good predictions. So one of the traits that's very high on that graph is branches per tassel. Now we know this actually does matter for yield. If you look at modern hybrids, they have smaller tassels with less pollen. If you think about a wild plant where there's competition for reproduction, you want to make lots and lots of pollen, outcompete your neighbors. We just need enough pollen that every grain is going to get one pollen grain. So we can shrink that tassel, less energy goes into the tassel, and you get less shading. So there's some really nice classical studies. If you detassel your corn, you will get higher yield if you detassel right after pollination, because you're not having that blocking of sun reaching the canopy. But there's another problem. This population includes lines developed over a really wide time range. So part of what that branches per tassel is telling us is more recently developed elite lines, XPVPs, have fewer branches per tassel. So it's leaking through whether lines are more recently developed elites or older lines. Still, the potential to go in and figure out what the breeders are actually looking at is really powerful, because we are never going to replace plant breeders, but we don't have enough. And we're never going to have enough. This is particularly true as we start thinking about expanding to more crops, more niche targeted breeding for specific regions. I want you to notice, I mentioned this previously, all of these traits are things that students, postdocs, occasionally professors, can measure walking through a field with a ruler and a notebook. And that's important. A lot of the data we have still comes from this, still comes from folks with the rulers and the notebooks. My lab has had the fortunate opportunity to be starting right as a huge amount of investment, a huge amount of innovation went into high-throughput plant phenotyping. And we've heard some examples where it actually is starting to substitute for current human labor, particularly in industry. But there isn't a lot of that. That doesn't mean that these tools aren't powerful. It means a lot of their power comes not from replacing what humans can do today, but allowing us to do things that humans don't do today because we're really bad at it or we're really inefficient at it. So let's give you an example. So this is work from Jensina Davis, a current PhD student in my lab, currently on the job market. If anyone is excited by this and the other times her slides or her photo is going to show up. This was one of her first projects. We worked with a team at Purdue University and developed a method to reconstruct the 3D structure of a corn or a sorghum plant from multiple side view images. The baseline algorithm is something called voxel carving, which is really widely used. The challenge is that plant leaves are very thin and they tend to bounce around a little bit. So even if you take your photos a few seconds apart, the leaf often ends up completely vanishing. So there was some innovation there just to actually get the 3D structure. But once we had the 3D structure, we could extract different measurements from it. One is leaf angle, which we know is very important for optimizing corn for a high density planting. But another is phyllotaxy. So if you picture sort of an idealized corn plant in your mind, it has one stem and it has leaves coming off from one side and the other in alternating pattern. Some varieties of corn, some varieties of sorghum are really good at that. Others are much more noisy. The leaves can come off at different angles. There's been very limited quantitative genetic investigation of this trait to the point we didn't even know how much of this was heritable and how much was just some plants are weird, because it's a huge pain to measure this trait. As part of this, we wanted to collect some measurement. Let's say this in a slightly different order. So we realized with this 3D reconstruction, we could extract the phyllotaxy of individual leaves and quantify that for individual plants. But we didn't know, were these measurements accurate? And so we did the thing everybody does in these cases, which is we said, well, we have to measure ground truth. And this is where we realized just how hard it is to measure phyllotaxy with ground truth. We tried different things. We had pieces of string dropping down to protractors. Finally, the best method that Jensina and other folks in the lab came up with was opening their phone using the Compass app and then lining the Compass app up with the midrib of the leaf. And then we could look at sequential leaves and extract the data that way. And so what you see on the, what is that? My top left, your top right. Anyway, that panel C is the results when we did that. So we had our ground truth data, we compared it with the computer. The correlation was terrible. It's discouraging. So at this point, Jensina had a choice. She could say, okay, our method didn't work. Let's walk away. Do we do that a lot? But she asked a different question. And it was hard because she was the one who did a lot of these measurements. How do I know it's the computer that's wrong? And that's what you see down there on the panel A, which is below panel C, not important. The comparison of two human beings. So Jensina, another student, went into the same greenhouse, one after another, exactly the same protocol, measuring the same leaves. And that's the degree of agreement if you have two human beings make the measurements. It's only slightly better than the human and the computer. So an awful lot of the error that we're seeing when we compare the human and the computer for this very hard to measure trait is just humans are bad at it. And there are a lot of plant phenotypes like that that we know are important, that we don't study because they're hard to measure, and that we cannot validate accuracy just from saying, well, how well do they match the human beings? Because the whole reason we don't study them is human beings are terrible at it. But this is a hard trait. I mean, I made the whole point of setting this up as this was a really complicated thing to do. What about something easy? So I work on both corn and sorghum. Love both my crops equally. But when I have a stakeholder who's not an agricultural person, and I want to convince them about plant diversity, I bring them to my sorghum field, not to my corn field. Because You can see the genetic diversity of a sorghum field in a way you can't in corn, in large part because you have these big exposed panicles. There are lots of different colors, different heights, things like that. And as part of some work we were doing with the Nebraska Food for Health Center, which is trying to figure out how the genes in a sorghum plant alter the gut microbiome of humans that eat the sorghum through controlling the production of different metabolites, we wanted to just make sure we could track down and map all the genes controlling variation in color in sorghum seeds. Sounds like an easy enough thing. Read a lot about the best practices. We had a color rubric. We had examples of the different colors in Petri dishes, so it wasn't just that one person's yellow, somebody else's orange. And what you see here is the agreement rate of two different students assigning the same sorghum genotypes to different color classes. And it's not good. So mostly, human beings agree on light versus dark. Beyond that, it's really anybody's guess. You will also note that there is at least one example on that slide of a genotype where one student said it was white and one student said it was black. Now, the charitable interpretation here is, we can have labeling issues, we can have planting errors. At the same time, we have changed lab policy. You're no longer allowed to have headphones on while you're scoring seed samples. In any case, then this is work from Nikki Shritha, another PhD student in the lab. Nikki took this data, but she also took data from flatbed scanners of scanning the seed and could extract the RG and B color values from the seed. The scanners were much slower than the humans. It did not save us time. It did not save us money. But what you see on the top is the result of a genome-wide association study using the human-scored data. And what you see on the bottom is the result of a genome-wide association study conducted using the seed scans. We have a couple of genes that we already know affect color in sorghum, including Y1 and two loci involved in the production of tannins. And we can recover at least two of those loci, even with the human data. But we get more statistical support for those with the scans, and we identify additional signals that we would have missed just relying on the human scans. So even for things where it seems like humans should be good, this is not a given. You have to test it. And often, even if the computer is slower and more expensive, you get much better data out the other side. So these were kind of toy examples. Let's talk about one that really happened in production just in the last 6 to 8 months in my group. So it turns out another thing humans are bad at is consistently scoring disease severity. There's a consistently that is missing there. And we heard this morning, a really good person can do a really good job scoring disease. They can do a much better job, I think, than current methods with images and image analysis. But there's this really great study that came out of Cornell 15-ish years ago where they had a real population of corn. And I forget what disease it was. Anyway, there was a disease infected on these plants, and they asked different people to go through and score the severity of the disease, same rating scale, same pictures. And what they found was that there were some QTL that agreed, but there were other QTL that popped up just if one person had done the scoring or just if a different person had done the scoring. The same people had looked at the same scoring metric, and they'd interpreted it differently. Now, within one environment, that's not such a big problem. And I would say we have recapitulated this work in my lab. This is work from Southern Rust, published a few years ago. We had a major outbreak in our field, and we had 3 people go to the field. And what you see here is the distribution of disease severity scores by those 3 different people, and then the degree of agreement when they scored the exact same plots. They're clearly seeing some of the same things, but there's a lot of inconsistency. So in one environment, this isn't so bad. You can just make sure you have one person that you really trust. They go through, they do all the scoring. As you start thinking about wanting to look at disease severity across many locations distributed across the country, it becomes much more challenging. Either you're going to put your most talented person on a plane for months and still probably miss some of the sites, depending on when the optimal scoring is, or you're going to get a lot of score-based variation that's going to look like G by E variation, because the scores and the locations are confounded. So we were talking about this problem with one of my colleagues, Sabyul Kim, a plant pathologist at University of Nebraska. And this is work both by Carla, who you see here, and then Jensina, who will show up on a later slide. And as it happened, that summer we had as we actually often do with this, we had a relatively severe anthracnose outbreak in one of our sorghum diversity panels. So we wanted to be able to score this in an automated fashion. We came to this realization in late July, and that gives about a month before the sorghum really starts drying down. So we decided to go really rapidly. But Jensina worked with a very talented technician in my group, John Turkis, went through a whole optimization process of how are we going to collect these images. Because one of the big challenges with many image analysis methods is you spend a lot of time after you get the images just trying to figure out, how do I clean this up? How do I get something that I can compare? So here we invested a few weeks on the engineering side, and it made the image analysis so much easier. What we ended up with is that box you see there. The leaf gets photographed always in a uniform environment, always with a clean background. It makes segmentation really easy. It really reduces the need for color correction. And it's fast. So with 8 people, we got about 6,000 leaf images in 2 days. And I think I have slides later on here. We could also ship this around to our collaborators in both Alabama and Georgia, so we could get consistent images from the same method. So this imaging produces a bunch of photos like this. What you can see, not all the leaves are the same. One thing we could have done now is we could have asked Sabyul, or one of the really skilled pathologists, to just click through 6,000 images and do all the scoring. And we may still do that. This project is ongoing. But we did want some sort of an automated method to extract this, particularly because we think the one axis of disease severity is probably masking a lot of variation. The reason different people give you different scores is they're looking at different things. And so if we can disambiguate those, we probably will do a better job of identifying the genes responsible. I'm not going to go into this in too much detail. This is, again, work from Jensina. But essentially, after trying several approaches, what we found was most effective was to take a pre-trained object detection framework, chop off the top layers, extract the embeddings from that framework, and use these as our measurements. So let's see, what you see here is a correlation between predicted disease area using these embeddings in a much simpler technique. You will see there are some outliers that are predicted to have very high disease severity. Our method using the object detection doesn't identify them. We could actually pull those out, and those were dead leaves. So really simple methods are going to call dead leaves disease. This method did better. We have a bunch of non-redundant encoding embeddings, and then we can do GWAS for those different embeddings. Don't look too much at that Manhattan plot because I sent these slides in Wednesday morning. Since then, Jensina has sent me an updated version that got much, much better. It turned out some of the names were not formatted correctly, and so we weren't matching the genotype and phenotype. It's always good to check that. And then we can extract from these individual embeddings, and we can start to look at what are the types of differences that the different embeddings are capturing, and then work with plant pathologists to tie that back to what does this actually tell us about what's happening in the leaf. Well, that's that slide again. All right. So up to this point in the talk, I mean, I've talked a lot about how to extract phenotype data from different approaches, from different automated methods that usually aren't faster than humans, but sometimes do things humans cannot do. And for essentially any process where we're trying to either make breeding decisions or identify genes, anything like that, we need 2 types of information. We need that phenotype data, and we need genotype data. Now, when I started out as a professor, almost every talk had some version of this picture at the bottom, showing you the rapid decline of sequencing costs. I think that now that's kind of a given. The point I wanted to make in this first part of the talk, though, is phenotype data is going to remain rate limiting for us for a very long time. So what we need to do is think about ways we can use that rapidly declining curve to compensate for the fact that the phenotype data will remain expensive. Okay, so it'll remain expensive. One of those is that there's still a lot of information that we can mine from previous field studies. Once you have put a huge amount of work into growing out a field population, collecting data, often you can come back and either with the addition of new genetic or new phenotypic data, you can learn new things from that same data set. So here's an example of just meta-analysis and merging together measurements conducted with different populations. So this is work from Ravi and Harshita Mangal, a current PhD student in the lab, with the sorghum association panel. This is a set of about 400 lines that have been used for GWAS, I think for close to 20 years now. You can go back through the literature and assemble something like 700 different traits that have been scored across this population. This lets you start to answer questions about pleiotropy of quantitative loci. But, and this is honestly one of my favorite slides in the talk, because it's just so simple. What you see here is an analysis, a genome-wide association study conducted for flowering time. In this case, we're back in maize. This is a population of about 750 lines. In both plots, this is the exact same phenotype data collected from the same field from the same plots. The only difference between the top and bottom plot is the top plot was scored with about 1 million markers derived from RNA-seq, and the bottom plot was scored with 40 million markers derived from whole genome resequencing. Just by adding more markers to the same population, we get a whole new signal. In this case, we have 2 genes, both of which we know are involved in flowering time in maize. So think about how many data sets are sitting in your lab that you've published that were analyzed with what was the best marker data set at the time. And how many, and my guess is that in many cases, there are new associations that you could pull out of those data sets today with the markers that exist, particularly for populations like diversity panels that get reused again and again. But this doesn't actually get us as far as I would like, because I've labeled these 2 peaks. We already know what the causal genes are. But if we zoom in, what you see here, the red triangles are the positions of the causal genes. The dots are showing us all the markers in LD with those causal genes. We don't do a very good job of narrowing down to single gene resolution with genome-wide associations, particularly in plants. And this is maize. Maize has relatively rapid LD decay. This is an example from Wakar Ali, another graduate student in the lab, working with a number of photosynthetic traits. What we would really like is examples like panel B. So what you see is the original marker that was identified as a colored dot. The red dots indicate everything in LD with that, and then the black triangles indicate annotated genes. So we have a marker, we have an LD window, there's 1 gene in the window. We don't know that that's causal, but we have useful new information. However, what often happens is instead you get something like panel C, where you've got the marker. There's really nothing that you can connect it to based on LD. You can maybe say, well, these are the closest genes. They might make sense. Or something like panels E or F, where you end up with 10 or 20 genes. And at that point, I would argue you really have not learned anything new, because saying this gene is one of 20 genes, one of which likely is linked to a phenotype, really doesn't give us a lot of new useful information. But again, we can use advances in our ability to generate sequence data to get around this problem. And specifically, what we can do is use something called a transcriptome-wide association study, which is essentially the same thing as a genome-wide association study, except instead of using individual markers, we're using the expression levels of genes. Now, this has a number of advantages. One is that it's much less sensitive to LD, and there have been a number of studies to show this. Another is that you're working with many fewer variables, each of which is relatively more information rich. There's a lot more information from the expression of 25,000 transcripts than 25,000 random SNPs. But it also has some major disadvantages. One, with genetic marker data, I can sequence a population of 300 today, and when I have money, I can come back in a year and sequence another 300, put the data together very easily. Maybe I'll have 900 in 2 years. That doesn't work with RNA-seq. To have comparable data, I need to look at plants grown in the same environment. I need to look at the same tissue. Really, I need to look at the same day, because there's a lot of environmental variation. particularly in the field, but even the greenhouse. And even on the same day, we know most transcripts exhibit diurnal cycling. So if I compare a sample in the morning and the afternoon, there'll be a huge amount of variation that is not actually driven by differences between the individuals, just driven by sampling time. So after some thinking about this, we realized if we wanted to do this right, we needed to collect samples from our whole population in a single day, flash freeze them in liquid nitrogen. We had to do it in about 2 hours. And so fortunately, we realized this right as John Turkis, my lab manager, joined the lab. This was during the pandemic. So he started, and I basically didn't see him for the first 2 months. He started, we had a Zoom call. I said, hey, we have this problem. By late June, we have to do the sampling. And he went away. And he came back with what turned out to be a really powerful system. And I'm going to, let's see if these will play quickly. So one of the things we realized is you're running through the field really rapidly. You have to pre-label your tubes. You don't have time to do it. So you've got all your tubes pre-labeled, pre-organized. It's very easy to kick the tray, everything goes flying, and you're out of luck. He came up with these spill-proof trays. Another thing we realized is no matter how good we are in molecular biology, sometimes RNA extractions fail. And unlike DNA, we can't go back and get another sample from the same plant at the same time. So the solution, again, was mechanical rather than AI. So John came up with an adapter to take 3 leaf punches, essentially connect them all together, so every leaf gets sampled in triplicate. And 3 tubes go into liquid nitrogen all at once. So we have 1 tube. When that fails, we have a backup. We've never actually had to go to the 3rd backup so far, but what that means is I have all of these populations where I've got hundreds of frozen leaf samples sitting in my -80, which turns out to be really good when you want to recruit collaborators working on metabolomics, working on cell wall composition, because the samples are all there. It's just immediately going to the analysis. And again, we have that layer of redundancy if for some reason we had 2 RNA extractions fail in a row. And when we take this data and we use it again to analyze the exact same flowering time data I've been showing you for the last 7 or 8 minutes, we get a lot more signals. So what you see there in the bottom is genome-wide association with 40 million markers. On the top, transcriptome-wide association with 20,000 transcripts. You will note that there are a lot more signals. A lot of them are genes that already make sense. And by definition, each of these signals represents a specific gene, not a window that could contain anywhere from 0 to 50 genes. So there's a lot more potential information content here. But this is still validation, right? I'm saying, look, we did this analysis and we found things that we already knew, so it works. I want to quickly tell you an example of a story where we actually learned something we didn't know before. So sorghum has a very different history from maize. Maize spent thousands of years adapting to temperate environments. It was originally domesticated in tropical environments, came to sort of the middle of Arizona and stalled out for a long time until its photoperiod could adapt to the more temperate environments. Sorghum had a much rougher transition. Grown in Africa, South Asia, came to the US, and over a period of a couple hundred years, was adapted to temperate environments with some very large effect mutations that affect photoperiod sensitivity. And we know a fair bit about the genes involved in photoperiod sensitivity in sorghum. But when you knock out photoperiod sensitivity, you unmask a lot of additional variation in photoperiod, in flowering time variation that is comparatively less studied. So we had this large population. We had both genetic marker data and transcript data that had been generated from it. The same principle as before. So here's the genome-wide association study. And we had all 3 possible outcomes you could imagine in a genome-wide association study. MA1, maturity 1 gene, probably best studied flowering time gene in sorghum. We found it. Chromosome 3, we found a hit that's right on top of an FT-like gene. So we found something that wasn't known before in detail, but something that makes a lot of sense. And then we had this wonderful hit in chromosome 9, not close to any gene that made any sense for flowering time. So we're in the same position of not having learned a lot that we didn't know previously. We validated some stuff. We found some stuff that makes sense. Yeah. How am I doing that? Yeah. When we do the transcriptome-wide association study on the same data, we get a lot of signals. But what was really striking is initially we got a lot of signals from genes that had no protein sequence similarity to anything else, even to maize. We spent a long time banging our heads against this until we realized these weren't actually protein coding genes. So if you are downloading your annotations from Phytosome, typically Phytosome really does not do a good job of capturing non-coding genes. These were microRNAs, particularly miR-156 that were showing up as associated with flowering time in our population. That, plus the signals from these SPB transcription factors, led us to the aging pathway, which is well characterized in Arabidopsis, linked to flowering time. And that, in turn, led us to miR-172, which is not showing up in this analysis, but turns out to be right next to, about 40 kb away from that signal we got in chromosome 9. Since this paper came out, we got access to a lot more flowering time data. We could do a lot more fine mapping. Now, the strongest signal we see is only about 2 kb away from that miR-172. How am I doing on time, actually? 3 minutes? All right, let's accelerate. I think we don't have to go through this whole story. Yeah, so we can do lots of eQTL work. We can do lots of cis-eQTL work. Yeah. So, no, that's not going to make sense either. You know what? Thank you all. Let's have a few questions, and yeah, thank you for your attention.
Thank you. For the TWAS stuff that you're doing, so are you using SNPs, or are you using, like, if there's transcript variation, isoforms? What are you using in that?
Yeah. So this is direct RNA-seq. So we're measuring the expression level, and we can convert and rescale that to a 0 to 2 value. So it gets treated essentially like a marker in the statistical models we would use for a GWAS. And we are using that right now at the level of 1 representative transcript per gene. We've experimented with looking at the separate transcript isoforms. With the current data, I think we're not getting enough resolution to really resolve the isoforms. If we move to sort of a longer read technology, we may be able to do that.
Hello, thank you for the talk. It was wonderful. So I see that you tailor your approach to the problem that you're trying to solve. But in general, do you believe that the takeaway from these experiments has been, at least for you, that you need to find some way to integrate genomic and phenomic data in order to get the best possible results, or am I incorrect?
Generally, yes. I mean, I'm sure that there are edge cases that you could get away with just really good genetic data or really good phenotypic data. I mean, there's lots of plant breeding that really is made just with good phenotypic evaluations. One of the big transitions that I'm seeing in my own group, and I think is happening more generally, is we are moving from a world where we have multi-million-dollar phenotyping centers that collect lots and lots of data, and then you figure out what biological questions you can address, to a world where we can rapidly develop a specific tool for a specific job that costs $50 to $100, and then go out in the field and deploy it. And that's a lot of fun. It gives us a lot more freedom to do that customization.
And if I may, just wondering, any advice to the students that design the symposium, if they are interested in how do you achieve, you work for a lot of companies, you do a lot of things, you publish so many papers. What is your key to success?
That's a good question. So, I mean, an honest answer, no. A slightly more political answer that is still true. One of the reasons that I look very productive is I collaborate with a lot of different people doing a lot of different things. And the reason I think I've been reasonably successful at that is putting in the time to think about what other people's incentives actually are and not assume that they're always the same incentives as me. The toy example of this that still wows me is I work with a lot of engineers that are in fields where the value for a PI is to be as close as possible to the front of the author list. And I work in a field where the value of a paper for me is to be as close to the end of the author list as possible. So we're not even competing for the same things. And it was really valuable to learn that. working with statisticians and finding out that actually coming up with an interesting new problem is already a valuable contribution to them, particularly if you're willing to put in the time to write an introduction on why that problem matters, things like that.
Thank you.
So you mentioned that you take them to the sorghum field because it has obvious diversity.
Yeah.
And my question is, with all that diversity, why haven't sorghum yields kept up with the pace of corn yields in terms of improvement?
Mm-hmm. Yeah, that's a very good question. I know of at least 3 answers to that, and I don't know what their relative contribution is or how much sort of residual isn't explained by that. The first is that there's been an awful lot less investment in sorghum breeding than in corn breeding. I mean, you look at the number of trials across the country. The second is that sorghum, almost all the sorghum we grow in the U.S. is grain type, and it has several really large developmental mutants that create the dwarfing, the sort stature, and bring the panicle down to about the same height as a maize ear. So it's easy to harvest with the same equipment. And it may be that when you have that level of change, it makes a lot of the standing genetic and phenotypic variation less relevant to improving yields. I said 3 reasons, right? Those were the 2 that I'm remembering right now.
I'm interested in your flowering time story.
Yeah.
So unlike the Arabidopsis, both corn and the sorghum have the female flower and the male flower. So
Yeah.
how do you use these 2 traits? And in your TWAS study,
Yeah.
do you see the genes or pathways that are very different from Arabidopsis?
That's a good question. So you said in maize we have separate silking and anthesis data. In sorghum, when we get one set of flowering time data. We have done separate mapping with sorghum, with silking and anthesis data. We get slightly different results. We have not been able to, so far, identify any loci that control what's called ASI, so the interval between anthesis and silking. One of the challenges whenever you want to look at a delta between 2 phenotypes is you get all of the error on this side and all of the error on that side. And similarly, with the variation we get in the hits for the silking and the anthesis data, I haven't convinced myself that that is biology versus different levels of scoring accuracy depending on who's walking through the field and how they're looking at pollen shed versus the emergence of silks. But it's a really interesting question.
What about the TWAS? Because you just basically measure the leaf transcriptome.
Well, that's the amazing thing, right? So this is not the tissue where any flowering time decisions are being made. And some of those strongest hits are ones that are functioning directly in the apical meristem. It's not signals they're not signals that are occurring in the leaf and moving to the meristem. So I think a big part of why TWAS is working so well is that we are capturing sort of global cis-regulatory variation. We have examples of using expression data from seedling roots and capturing genes that are involved in varying endosperm composition. So a lot of this variation, I think, is not tissue specific. It is global up and down regulation.
Thank you.
Yeah.
I was just going to ask you with the flowering time for sorghum and for maize, you both presented.
Mm-hmm.
How common are the pathways between the 2? Are you seeing? You didn't really comment on whether or not it's the same genes or not, and can you use one to find variants in the other?
No, that's a good question. So of the hits that I showed you, so the aging pathway does affect flowering in maize if you knock out, if you have a corngrass mutant that has very different flowering, it stays in juvenile vegetative for a long time. There isn't a lot of evidence for naturally occurring variation in that aging pathway in maize, as far as I know. The maturity 1 gene that's involved in sorghum's photoperiod sensitivity, as far as I know, that hasn't been linked at all in maize, but a lot of the work in maize occurs in long temperate adapted lines, so it's quite possible that a gene involved in photoperiod sensitivity wouldn't fall out of that analysis. The FT-like gene that we identified in sorghum is the direct ortholog of ZCN12, so not ZCN8, which is really probably the most widely characterized one in maize, but there is a fair bit of suggestive evidence for maize that ZCN12 is involved in determining flowering time in maize under photoperiod inducing conditions. So normally we wouldn't pick that up in trials conducted here in the central in the U.S. But if you get tropical data, it looks like that one does matter. So that's probably the best example of conservation between the two. All right. Thank you, folks.